I'm Bob Martin. I'm moderating the session. I'm going to introduce all 5 speakers at the beginning, and then they're just going to go one after the other. We'd like people to hold questions until we're finished. We'll have a question and answer session at the end. And before we start, I do want to make the announcement that we have a Red Blotch webinar next Wednesday, 12 till 1:30. And we also, that the ODA has a project funded through APHIS to do some red blotch testing. And Deepak Pudo from ODA is here, and they have capacity to do about another 100 samples on this cycle of funding. So if you have suspect vines that you know, they can take testing now, or we can pull leaves in May, but you won't see symptoms between now and the 1st of June. But the sampling needs to be done by then. Okay, so the session today is on red blotch virus. We have 5 speakers. We're going to start off with Dr. Sudarshana from UC, located at UC Davis. He works with USDA ARS. He's located on Davis campus. And then Dr. Frank Zalom from UC Davis, Department of Entomology and Nematology. And then Vaughn Walton from the Horticulture Department, horticultural entomologist here at Oregon State University. And Rick Hilton from the Southern Oregon Research and Extension Center, again, Department of Horticulture. And they're going to be talking about vectors in Oregon, the work they're doing. And we're going to finish up with Daniel Sweeney, who's a field representative with grower experience dealing with red blotch in Southern Oregon. And I think for a lot of people, the grower experience may be the thing you want to hear the most, what they've been trying and what's worked and maybe didn't work. So without further ado, we're going to get started. So we have time for questions at the end. And we'll start off with Sudi.
Good morning. Thank you, Bob, and the Oregon Wine Commission for inviting me to give this presentation. I started my journey with red blotch almost a year when I started my ARS job. That's in 2008, about 9 years ago. So I'll give you a little overview of everything what we have done so far. Okay, in the year 2008, when I started, we went to look for Syrah decline disorder in one of the vineyards in Sonoma County. What drew my interest was a Malbec block right close to the Syrah block, where we had symptoms where it was like leaf roll, but it looked different to me. And I asked my 2 experts who were with me, Jerry Uemoto and Adib. Well, they said, oh, it's leaf roll. Well, I took a picture and just kept quiet. Then same day, we went to another vineyard, Cabernet Sauvignon. Again, it's the same thing. They said leaf roll. And this had been already tested. It was negative for all the leaf roll viruses. And okay, I took a picture and kept quiet. The next year, in summer, Jim Ulpert, viticulturist at UC Davis, and his staff, Mike Anderson and Jason Benz, who was the manager at Oakville Station, they came to me and said, we have a problem. There is leaf roll-like disease, and the symptoms, the grapevine's showing symptoms every year. It's kind of increasing. We don't know what's going on, and they're all testing negative for leaf roll viruses repeatedly. And the important thing was they have reduced sugar and delayed harvest, just like leaf roll too. And it was not like one vineyard where we had a problem. No, there were 4 vineyards, all within 3 miles radius, having the same problem. So first I wanted to, because I was a beginner to grapevine diseases, although I had worked on viruses for 15 years before that. So I just try to ascertain what are the likely red leaf diseases. We have abiotic factors, tie tape, injury, and nutritional problems. Then we also have biotic factors caused by bacteria, crown gall, pest disease. I'll show some of the slides here. And they all can cause red leaf diseases, and sometimes, even now, I see people calling me to look at red blotch, but it's not red blotch, it's something else too. Okay, here is a tie tip related issue where one colon is showing red leaves, the other one is not. And there's a couple more like that. Then we have potassium deficiency, showing kind of reddening, which gets scotched later. Crown gall here causing red leaf, and pike anchor on the other side. Sorry if I use a pointer on only one side, I can't switch around, so I just try to point out on this side. Okay. And then we have paste disease, where the symptoms here, those who have red blotch, it kind of, the leaves can show symptoms similar to red blotch, but paste disease is quite distinct. Okay, this is a problem we have in California where it's an incompatibility issue, where we have some unknown agent. Bob is also working on this one for several years on Pinot Noir clones, where we have a union disorder where the whole canopy turns red. This is classic leaf roll symptoms, you would see. But when I look back at red blotch, see the symptoms were kind of different. You have the reddish veins, this on Cabernet Franc. This was 2009, before we saw the virus, okay? And then the symptoms were developing from the the leaf margin and these blotches. And this was not like a typical leaf roll symptoms where intervenal region is red, where the venal, the veins kind of remain green. So we started looking at what's happening here because we have at the time we had the new technology available, deep sequencing. So we looked at all 3 samples and tried to see what's out there. What we found was a new virus, a brand new one, which nobody had known at that point. It's a DNA virus. Most of the viruses then known in grapes were all RNA viruses. So because of the symptoms, we call it grapevine red blotch associated virus. Then the question was to see whether this virus is actually associated with the disease, clearly. Luckily, we had 3 isolates that we sequenced in the beginning, so we were able to establish there were 8% difference at the genome, hence 2 clades. Some of you, if you have heard there are 2 clades now, is because we had this covered right in the beginning. So we developed the primers, and then we started looking at all those vineyards where the symptoms were apparent. And we could easily see a PCR product in all the vines that showed red blotch symptoms. So the primers we designed in 2011 was robust enough, and we distributed that right away. A year later, a similar virus was found in New York, and then in one of the meetings, the international meetings in Davis, when I met Bob and others, I distributed the primers to everybody who ever asked. And then eventually we came to know that this virus was present in most of the vineyards where leaf roll had come negative, and that includes almost all the wine grape regions in the country. Okay. So going back to the symptoms in different cultivars, this is how it is in Cabernet Franc. And this is to show the contrast between symptoms between red blotch and leaf roll. You can see the green veins here clearly also. Whereas in case of red blotch, we see the red veins and the blotches. But this is not always like a done deal. These symptoms are not always the same across the different cultivars. This is in Cabernet Sauvignon. It's a little different. And this is a second leaf vineyard where 95% of the wines had already, they had the disease in second leaf itself. This in Malbec. Even though I went to a different vineyard in 2013, it was just exactly the same as what I had seen in 2008 in Sonoma County. This was from Napa County. Okay, this in Merlot. And Pinot Noir, I think most in Oregon might have seen this one on the Pinot vineyards, the red blotch symptoms. This one can be confusing. This can resemble more like leaf roll to start with. Okay, this is a kind of a dry farming Zinfandel vineyard, in Amador County. In the last 2 years, because of drought, the vines with virus were pretty much beaten up. This is in Mendocino County, another Zinfandel clone. The symptoms are not that obvious in white cultivars. This in case of Chardonnay. And you can see it's more like a some kind of a nitrogen deficiency or something like that. It is in white Riesling and in Rizon. And this also kind of resembles the potassium deficiency we would see in some vineyards. Actually, UCD, the website, they have symptoms of potassium deficiency that looks exactly like this. Okay, so getting to detection, the primers we developed in 2011 were very good, and they're still in use. Although there is, we have newer techniques. This is by using conventional agarose gel electrophoresis. You can see all the symptomatic vines showing the bands. It's the same slide as the previous one. And then we are also developing, we have developed real-time assays too, using Tachman probes or dye-binding, DNA dye-binding dyes technique. Here, what I'm showing you here is a new detection technique we are developing. This is by using isothermal amplification, where we don't need heavy instrumentation. Simple, about $1,000 investment is good enough. And the cost is expected to be about $4 to $5 per sample. That's what we are aiming at. And here you can see, it's a simple extraction method where we can put the tissue in a buffer and boil it for a minute and take the extract without any further manipulation and use it for a PCR. The extract what we made, even if you dilute like 5 times, 10 times, 50 times, 100 times, it can still give us positive. So this is with the infected tissue and the healthy tissue. And the same products, we can run it on a gel and see a band too, here. And we can take it to the lab on instrumentation, and we can also do high resolution melting analysis to make sure the DNA products what we got is from red blotch itself, red blotch virus. Okay, so a little bit about the impact, what we have seen with red blotch in North Coast. And the Cabernet Franc vineyard, the healthy or green wines, here the clusters had been removed already, and this one had to wait for 3 more weeks before they could pick the clusters. In North Coast, this can be a problem. If they lived longer, we get rain, and then we get other problems too. Then, this is our second year block, and this vineyard had planted in 6 acres, and they ripped off this one when it was 4th leaf. They had to write down $1 million loss for the 6 acres. And so before they ripped off, they asked me to test some wines and made sure which one had red blotch virus, which one didn't have. And they made wine, they analyzed the juice and also made wine later. Those which had red blotch, they had nearly a drop of about 5 to 6 units in bricks. That was a big difference, and because they need to wait for like 26 bricks at least to harvest it for winemaking. And then the titratable acidity was higher with red blotch, and anthocyanin was low. In a year later, the same wines were again analyzed, but this time they kind of bulked it. They did not go individually. The difference was about 2 units in the following year. So on different years, with the vintage, you can have some differences in the bricks. It's not always the same. And that winery, it's a high-end winery, they ran their data through an economist, and what they found is if they were selling their bottles $100 per bottle, they were going to get $90,000 per acre. With red blotch, their revenue was going to be only $10,000. So one of the problems they had was, in the beginning, convincing nurseries and everybody about the red blotch effect. It was not easy for the growers to do that. So based on the data I had, they made wine and they brought people, nurseries also, to taste the wine and feel the difference themselves. I don't drink wine, I'm sorry. I'm just non-alcoholic. But I can smell the wine. I can tell the difference which one has red blotch, which one doesn't, which one didn't have red blotch. Of course, I drank wine later. Here is the vineyard where, until like 2012, it was kind of clean. There was nothing red. And in 2013, some of the wines started showing red. 2014, there were more that were red. And what I believe this was hit by a vector carrying the virus. That was our first evidence to say that something was happening there. And here is the, when you go and take samples for testing to a lab, often you try to make a composite drawn like one cane or leaves from different wines. Here's a case where you have two cordons. One cordon is showing red, the other one is not. We took samples at each of the bud spurs and ran the assays separately, and all those with red came positive for the virus, and this shoot close to the red, this cordon also came positive. This one did not. So if you took sample here, it would be negative. If you had taken this sample here, it will be positive. Okay, this is another case. This wine in Oakville Station, year after year we had tested. For 3 years, it was negative. In fourth year, you see this single shoot showing symptoms? That had red blotch in that year. It was a new infection. So we mapped, actually, this is one of the blocks which in 2009 first we looked at. And then in 2011, once we got the virus, we tested, made sure the virus was present in all the symptomatic wines here, and the asymptomatic wines, by and large here, didn't have the virus. And we monitored the percentage of infection. In 2011, it was 46%, and it went up to 63% 4 years later. This block was also ripped out, and we lost our site for further work. And what is interesting is, we had one clade that was predominantly present in the 2011 and 2012 samples. Now, 11 is for 2011, 12. Then 13 and 14, we got another clade that came from this direction. Okay, so at this point, I decided I will invite Frank to join me for studying the spread. And all the vineyards which I looked at from 2008 to 2013, they were all gone. They were all ripped out. Okay, I'll stop it there with just the one message. The people who said they had red blotch, they went through 4 phases: surprise, anger, denial, and then karma. Okay.
Let's see. I don't know if do you start mine now? There it is, okay. Yeah, my name's Frank Zalom. I'm from UC Davis, Department of Entomology and Nematology, and that was a good point for Sudi to leave off, because he wanted me to go see some of these vineyards that he'd been working on, and like he said, a lot of them have been removed already. And I remember Sudi telling me he feels like the angel of death, because whenever he's working in a vineyard and he says, he actually does the PCR work with the vines and everything, a year later or 2 years later, there was no place to work because the vines were gone. And I started realizing that after a while. So, Sudi convinced me that there was something going on beside just the nursery transplants that had gone out years before we were able to identify that there was a virus. And I think that, at least in California, my feeling is a lot of the vineyards that are planted where we have 100% infections all probably were planted with infected nursery stock before we knew there was a red blotch virus. But we had these situations where you could see these patterns of spread, where there was no other way to explain why we didn't see symptoms for years, and then all of a sudden we'd see individual vines, and it looked like the vines were increasing. And then there was some pretty convincing evidence where we'd actually mapped vineyards that from year to year we were seeing the number of vines increasing, and these were confirmed with PCR on the individual vines. And then also, this is purported to be a Gemini virus, and all known Gemini viruses are insect transmitted, which would make you believe that there must be a virus involved in this. So this is just, I'm not going to go through the data because Sudhi already presented this, but this was the block that we had at Oakville Station, the University of California Davis's field station in the Napa Valley. And basically, he's already shown that, when we started working in that vineyard, there was about 44 to 46% infection, and then it increased by 4 to 5% a year. And all of the vines between the two black lines on there all had PCR done in each of the years, so we knew if the vines were positive or not. And so that was, pretty convincing evidence. The reason why it only goes through 2014 is because they pulled the vineyard after that, and that was a decision. And I really wish the vineyard was still in there so we had a chance to keep doing the research on it, but we even lost it at the university. But as we went around the state, we saw a number of other sites, and it wasn't just in the North Coast. This is in Santa Barbara County, so in that Santa Maria area, but in the northern part of Santa Barbara County. Now, the interesting thing with this is that this had been a vineyard that was planted, and I'm not sure exactly the age of the vineyard, but there were no evidence, symptoms of red blotch for many years. And then there was a vineyard that was planted immediately adjacent to it that was 100% infected. And so that was probably a vineyard that had been previously infected. The nursery stock was probably previously infected and it was planted out. And then you can see the pattern here that from the border from where the vineyard was planted, we started seeing infections. And we've gone back now and mapped this in subsequent years, and we've seen the number of vines that are infected increase, and have been able to show that it's coming from that side of the vineyard. And we've seen this in a number of other areas as well. And so people were saying, well, is it possible that this was these were infections that had been there for years and we're just starting to see the increases? Well, it's possible, but we ran into a vineyard in Amador County that came from self-rooted vines that were from a vineyard that was originally planted by the family in 1928. And that 1928 block is still planted. And then the family is like in its fourth generation now, and they've planted vineyards right around in the same area from that same mother block. The mother block didn't have any red blotch symptoms in it until we started seeing it in the last, I think, year or 2 years, where there's 1 or 2 vines in there that are starting to get it. But so this is a little graphic of that. And I guess I'm working on a Mac, and so when it goes over onto here, these are just block numbers, block 1, 2, 3. It's not $1, $2, $3 blocks. But the source of the cuttings were all that 1928 block, and I'm going to do the same thing as Sudhi did here. But the mother block was out in this area, and then they had planted several other blocks. There was like a little riparian watercourse that went between here. Then they planted this block, and this was like sometime in the late 1980s that was planted. And then this was the most recent block that was planted here. And this was planted I think it was right around 2000. And again, no, they had not seen any symptoms. And Sudhi actually has done PCR on all of these vines for all of the grapevine viruses, and there's no other viruses in there that we know of. At least nothing that's a known virus at this point. So that's kind of interesting. Then sometime about 9 or 10 years ago, there was a vineyard that was planted next to it here by one of the neighbors that is 100% infected with red blotch. And then all of a sudden, we started seeing, again, red blotch along this border, and now we've been going back and looking at all the vines every year. And usually we don't see a big increase in the numbers of vines that are infected, but in this particular case, we did have an increase in 1 year of 18% from 2015 to 2016. So you can see cases where something happened during that point where the number actually increases. But we really believe that there was a vector that moved it from this because you have to have, I was telling people at breakfast this morning, being an entomologist, the vector is really exciting and important, but you have to have the virus. The virus is what's really causing the problem. And if we don't have the virus present, you can have vectors present as much as you want, it won't matter. But that was a case where the vector wasn't present in the area. And a lot of people at that point were telling us before that time that in Amador County there was no red blotch. And then now that we've shown it in several vineyards and they're starting to get things set, so we know there's red blotch in Amador County, and this is one of the foothill counties in California. Okay, so is an insect involved? Well, in 2013, there was a study that came out of Washington State University that implicated Virginia creeper leafhopper as a vector in a greenhouse study. And so a lot of us that had been working at red blotch immediately started looking at Virginia creeper leafhopper, and we tried to repeat the transmission work, and this was tried in New York State and at least 2 laboratories in California. And I think, Vaughn, you started doing some transmission work, and none of us have been able to replicate that work. And so we still started looking for other possible vectors. It was a nice story because we saw large numbers of Virginia creeper leafhopper, but in a lot of these vineyards that we were looking at, we didn't see spread that was occurring, even though we had high numbers of the Virginia creeper leafhopper present. We also had vineyards where the leafhopper was not present, but we still saw this spread occurring. And the other thing was Virginia creeper leafhopper is a mesophyll feeder, and all of the monopartite Gemini viruses are phloem limited. So it would suggest that it should be a phloem feeding insect that's causing this. So that was a little inconsistent with that original diagnosis. So in terms of our transmission experiments, we started doing them in 2014, and we were just collecting every type of sucking insect you could see out in the vineyards. And we had done about 100 transmission bioassays, and these were all replicated probably 5 times or so. So we've used hundreds of vines to do this work. Vaughn knows that because he's got hundreds of vines in Corvallis right now too. So we did about 100 in 2014, and we had no virus detected by 2015. So we did this again with 125 transmissions. And added some different insects to that. And so some of the results that we had, if you look at the 3 erythranura, and this is western grape leafhopper, variegated leafhopper, which we have in California, and it was introduced probably 30 years ago or so and became our dominant leafhopper in parts of California. And then Virginia creeper leafhopper, which we really hadn't seen until more recently. We looked at all 3 of those species, and what was interesting is if you just grind up the insect, you do detect the virus in them, and that's probably because they're feeding on it. It's still in their gut contents when you're testing it, but they were found at very, very low levels. Just having it in the insect doesn't mean it can transmit it. But interestingly, some of the other hemipterans that we were looking at, and in this case, Cispistolis, and I'll talk about that one in a minute, had much more positives. So we were getting like 75% of them that we were collecting that had the virus as opposed to the leafhoppers. The other interesting thing was when we looked at the virus concentration, and these are CT values, so the higher values means the lower concentration. We had very low concentrations. As a matter of fact, very low concentrations with any of these leafhoppers, But with this Spicillus and a couple of the other hemipterans we were looking at, we had very low CT values that were very similar to what we'd have with an infected grapevine. So at the end of that period, we had, after a year and a half from the first inoculations with the leafhoppers, we had 0 positive. We did that second set, included the leafhoppers in them in 2015, and after 5 months, we still had 0 positive from those next ones. So we started really, at that point, agreeing that probably Virginia creeper leafhopper was not the vector that we were thinking about. And at that point, this was the end of 2015, we had a short list of species we were looking at. One was a tree hopper, two were leafhoppers that were phloem-feeding leafhoppers, a psyllid and a succeid. Okay, in February, we started getting some positive PCR results from our transmission experiments. And we found that at that point, we had 3 of 15 of the vines that we tested with membracid, which is a tree hopper, that were starting to come positive. And this was the little Spicillus that I talked about in that previous slide, and 3-cornered alfalfa hopper, and that was the tree hopper we were looking at. And this increased then as we were moving through doing the kinds, and subsequently Mark Fuchs, who's also been working at this from Cornell, also had transmission with 3-cornered alfalfa hopper, which made us feel better. And also, some of the vines that we had tested originally out of those 3 original vines actually started showing red blotch leaf symptoms. So you can see the mild symptoms here of red blotch on the leaves, and we didn't have these in our negative controls. Now, interestingly, and this was interesting as an entomologist, we did coat protein analysis of a number of different Gemini viruses, and we wanted to associate them with what their vector was. And so these trees show the relationships. The ones that are all in here are very closely related. They evolved sort of together. This is another group of Gemini viruses. These are leafhoppers. So all of these Gemini viruses are leafhopper transmitted, all phloem-feeding leafhoppers. These are whitefly transmitted Gemini viruses. There was one in the literature that was transmitted by a tree hopper, and that's tomato pseudo-curly-top virus. That's kind of a tropical virus, and you have it in Florida. And this is grape red blotch associated virus, and that's the closest virus to grape red blotch. And so it kind of lent a little bit of credence that maybe the tree hoppers indeed were, and we had this before we got the transmission, but it kind of suggested that tree hoppers might be a vector because it's so closely related to its sister Gemini virus. So this is the species that we had transmission with, 3-cornered alfalfa hopper, and these are the adults. The life cycle is they have an egg that they insert into woody plant material. They have 5 nymphal instars, and then they finally emerge as adults. The little nymphs are very characteristic because they have these spiny spines on their dorsum, and these are actually photographs that we had. So they're described as being wedge-shaped and having these heavy spines on the nymphs. Their eggs are inserted. So these are photographs that we took. They kind of make slits. The females make slits in the material and then insert the eggs. And so the eggs are really hard to find, but you can find the slits as well. The host preference, they prefer legume hosts. They have a number of other host species. The literature says that they overwinter in plant tissues or as adults protected by clumps of grass. And the type of damage that they cause are girdling. So if we look into publications where they describe girdling, this is girdling on soybeans. Again, leguminous hosts. This is alfalfa hay girdling. These are girdles that we see in grapevines. And this was actually taken before we knew that it was a potential vector. Do they oviposit on grapevines? Yes, they do. We cage them on grapevines, and here you can see the girdles from the grapevines. These are where they've inserted eggs, and these are the little nymphs that have emerged on the grapevines. So we know that they can survive on grapevines. We don't think that they go through their life cycle on grapevines. We've never been able to rear them through to adults. So we think that after the nymphs, after they emerge, they probably drop onto other types of hosts. We've only had 1 year of experience monitoring for them. We know they overwinter as adults. You can see this is March. In this case, we were sampling the cover crop in the vineyard. After the cover crop was mowed, we had very low numbers, and then we started seeing the populations increase through the year, and we were able to collect them all through the year. This is where I'm going to get into the next presentation. Are there other vineyard tree hoppers? Well, we didn't know of any other vineyard tree hoppers that might be, that really occurred. And we didn't, but we got this photograph from one of our extension advisors in Napa County. And these are tree hoppers that are all lined up here feeding in a vineyard. And these ended up being, they sent it to me and said, "Is this your tree hopper?" And we looked at it and we said, "Doesn't look like our tree hopper." And we went out there and collected them and tried to see what they were. And it turned out that they were a different genus called Tortistellus. It doesn't have a common name. This was the vineyard it came from, and you could see that where we collected the Tortistellus were up here on top of the hills, and they had red blotch up here, they had red blotch down in the bottom, and in the bottom they had Suspistillus, the three-cornered alfalfa hoppers, and on the hillsides they had Tortistellus, and so we hadn't really noticed that before. These are the Tortistellus species. We find both of these in California, and you'll hear more about that from Oregon here. One is Tortistellus, we think, wickami, and the other Tortistellus is Albita sparsus, and it's got little horns on it, where this one lacks the horns. They're much larger than our three-cornered alfalfa hopper. So this is the Tortistellus. This is three-cornered alfalfa hopper. And, we're just continuing with research on the tree hoppers now. And there's a number of different things we're trying to do with them. We have a vineyard that's planted at Davis that is in the second year now. These were all virus-free vines that were, and we've introduced the tree hoppers into these cages at different times during the season to see when they're infected. We've planted alfalfa on this side of the field. The alfalfa hoppers found it by themselves. We didn't have to infect it. And there's no alfalfa planted anywhere nearby. And then when we went back and took girdle data in here this year, by September, we planted the alfalfa in July, and by the end of September, we had 72% of the vines with girdles on them. And so it's really interesting how they can get in and find this, but we have no transmission data from this block yet. And so we're looking at a number of different types of management techniques. We'll probably have a chance to talk about some of that later. So I acknowledge all the people that have worked with us, as the nurseries and the growers that have worked with us very closely, our collaborators, our funding sources, and all the people that work in our labs. So I thank you for the opportunity to be here today. I'm sorry to be a little bit slow, but thanks.
Thank you, Frank and Sudi. I really enjoyed both of those talks. So I'll give a bit of an overview of what we're finding here in Oregon. We did some work in the vineyards itself, looking at the spread of the virus, and then we were also looking at the vectors. So with the new revelations about Tortistellus festinus, we were searching for Spicistolus festinus endlessly this past season. Rick was fortunate to find some in southern Oregon, but we didn't find them. I won't talk about the bugs, I'll talk about the virus. This project is supported by the Oregon Wine Board. We're also getting money from the Oregon Wine Research Institute. Frank, Zalom, and Sudi has really helped us tremendously to figure out the spread of the virus within certain vineyards. Before I start my talk, in a few months from now, on April 6th, we'll have our Grape Day. We'll talk a little bit more about these viruses. and give some more details. We'll have more time to give some of those details at that meeting on April 6th. And then next week we have that webinar as well. So what I'll talk about is what we call the deadly triangle. I'll show you which areas we looked at this virus, and then I'll share some of the epidemiology trends. Rick will talk about insect vectors and then best management practices. So this is that deadly triangle. You have the host, which we now know for the virus is grapevines. We have the pathogen, which we now know is red blotch. And then very important, you have the environment. And we're seeing that the environment's playing an extremely important role. We don't have too much time to go into that. But, and then, of course, the vector itself as well. These were the three areas that we were looking at. We were looking in southern Oregon with Rick and several other people who was working down there, Andy Swan and others, the Willamette Valley, and then the Columbia Basin. So I'll show a few tables here which shows the distribution within the different areas. But before we get there, I just want to talk about the methodology that we've been following. We've been collecting hundreds of plant materials within the vineyard, and then we were also collecting plant materials outside of the vineyard, including these little seedlings. And this brings me to the next slide here. Very important what Frank mentioned is you very often see this edge effect. We know in this vineyard where we took these seedling samples, there were red blotch in that vineyard itself. Went and took some seedling samples. And so when you're looking at those seedling samples, we found, these samples are more or less a year old, have them tested, and we're finding some of them are coming back positive. And this is the second year that we've been finding this. If we're looking at the different regions, we're finding a very wide range of infection, anything as high as maybe 73% down to 0%. All our nursery samples came back negative. We're finding it in southern Oregon, and we're finding it also in the Willamette Valley. If we're looking at an industry-wide number, and this is not necessarily representative of the industry in Oregon, that number might be lower, but the vines that we sampled, we found approximately 16% of those vines containing the virus. That's out of about 700 vines. So let's look a little bit closer. Let's look at some of these vineyards, some of these spread that we've been seeing from Frank and Sudi. Are we seeing that in the Willamette Valley? Are we seeing it in southern Oregon? And again, this is not necessarily representative of the industry as a whole. These are vineyards that we were specifically looking at where we were perceiving that there's spread taking place. So what we do is we would take those samples, we would have them analyzed using PCR, the same vines year over year, and we're finding this increase in spread. So in this vineyard in the Willamette Valley, we're finding 35% in 2013-14, and jumping up to about 60-some% in 2016. That's one scenario where we're finding approximate doubling of virus over that period of time. In southern Oregon, maybe this is the worst of the scenarios that we're looking at. In year one in 2014, 5.7%. I remember we had this grape day, we had 3 or 4 vines that I knew had the virus in, and we were going to walk to those 3 or 4 vines. We got to those 3 or 4 vines, and everything around it was red. So 2016, 62% infection, significant increase of the virus year over year. Another scenario, a little bit further away, maybe 5 miles, 6 miles away from that exact same spot, we're finding some spread, but not at that same rate. 14.5% infection in year 1, 2014, not as dramatic as in this other vineyard. And so this, in my mind, gives a little bit of a representation of how vineyards can differ from each other. If we look at the virus within the vineyard itself, there's eastern Oregon where we're at this stage finding very little. If we're looking at a vineyard distribution pattern, initially we were looking mostly at vine leaf roll virus. 1 minute left. In that vineyard, The bottom line, what I want to show here is that the distribution of those two viruses are significantly different. We were looking at symptomatic vines in 2009 and 2010 and so on, where they all came back negative. When we got the test, when Sudi started doing the test for us, suddenly we're finding a really good explanation as to what the distribution looks like. But really important here is that if you look at the location where the spread is coming from, it usually is from the edge of the vineyard, and then as the years progress, that distribution spreads deeper and deeper into the vineyard, ending up at about 60% year over year as you're looking at that distribution. This is significant clumping of virus using statistical programs showing the gradual spread year over year. I'm not going to be talking about vectors at all at this stage. We have found some presumed vectors. We have hundreds of plants in greenhouses where we're doing some of this transmission biology work with some of the insects that Rick will be talking about in the next few slides.
We need to go back to that one. So yes, Vaughn is having me talk about the insect vectors. He's leaving the fun stuff for me. So I am going to take a quick tour down memory lane if the slides come back up. But we have known that, actually need to go back to Vaughn's talk, I guess. It was combined. But we have known of the three-cornered alfalfa hopper, certainly in southern Oregon, as being a very minor pest in vineyards for some time. And yes, here we go. So this was a chapter from 2003 Oregon viticulture book I helped author, and just reminded me, we had it pretty easy back then. I mean, there was some grape phylloxera up here. There was maybe leafhoppers down in southern Oregon once in a while, but really everything was a minor pest. And we listed three-cornered alfalfa hopper as one of the very minor pests that you would find or run across. So when we heard the news that three-cornered alfalfa hopper was a vector, I sent this slide to Frank and Brian Botter. I said, we've seen three-cornered alfalfa hopper up here. Here's a slide I have from an outbreak in pears a few years ago. And Brian sent a very nice email back and said, we don't think that is the three-cornered alfalfa hopper. So that's this related species that we now think is Tortistolus wickemi, and that was sort of my first introduction to these other tree hoppers. So we did some surveying, Vaughn alluded to, and we also went back and searched all our sticky cards. We've been out looking in vineyards since 2009, starting when we were looking for leaf roll virus issues, and we also went through the collections, the insect collections from OSU. We have some collections in Southern Oregon going back to the 1920s. I went over to Southern Oregon University and checked their collections. And what we found were these three species that jumped out: Tortistolus wickemi, Tortistolus albidosparsus, and Spicistolus festinus, the three-cornered alfalfa hopper. And what you'll note is sort of a difference in where we find them. So Southern Oregon, we have all three species show up. In the Willamette Valley, just the two Tortistolus species. And in Eastern Oregon, really we haven't found one yet. They haven't shown up so far. And then when we look at where they're the most predominant, in Southern Oregon, really the Tortistolus wickemi is the major species. And if you look in the Willamette Valley, it's this other Tortistolus albidosparsus. So a real difference in the distribution. These are some glamour shots. This is Tortistolus wickemi. You can see, very handsome insect. The tree hoppers are noted by this really enlarged pronotum, and I think Frank showed the difference. So with our Tortistolus, it's a much higher sort of forehead, and they're larger than the three-cornered alfalfa hopper. This is the albidosparsus, the one that's found mainly in the Willamette Valley, and the sort of group in general is known as the buffalo tree hoppers, and you can sort of see some horns there on the pronotum. And this is just a top view. You can see the horns a little better. And also, this reminds me, you'd think an insect like that would not be a very good flyer. These guys can actually fly pretty well. They can definitely move and move around, and they're pretty active. So we did a number of different sampling methods in Southern Oregon. Sweep nets, sticky cards, beet trays, and interesting, we've only found one insect in the sweep net, a three-cornered alfalfa hopper in May. Nothing else. The torticellists just don't show up in the sweep net samples. Sticky cards, again, one specistulus, a few of the torticellis wickami, a couple of Obadisparsis. They actually showed up in some fruit fly traps in an apple orchard. And then the beet tray, not a whole lot better. We know you can find them with a beet tray because most of the insects in the collections come from beet tray samples. But really, the visual search was the most productive way we had of finding these insects. And again, you can see in Southern Oregon, a lot more torticellis wickami than either the specistulus or the Obadisparsis. So yeah, visual search. You're out looking for these guys. Even though they are, over a quarter of an inch, they're still a little cryptic, but we do have a cheat. We do sort of home in on the symptoms. So you look for that girdling, and here's that, the girdling they cause, and the torticellis wickami up above the girdle, and when that happens, as Frank alluded to, you get this discoloration of the leaf. Of course, you're only going to see that really in red cultivars. So we did do some testing where we caged leafhoppers on vines in a vineyard that did not have any red blotch, and so we did red cultivars, white, and you do see that reddening. It happens fairly quickly within a week, and of course it becomes more pronounced as the season progresses, but it doesn't happen in every case. So you don't get it, just because they fed on it, you don't always get the reddening. A lot of times there's partial girdles, not complete girdles. So a little test that Vaughn did in the greenhouse, they only had 1 out of 5 where you saw that real reddening from the feeding activity. So now that we know what the symptoms look like, we can go out and look for the symptoms and map those. So using the same statistical analysis that Vaughn used for his looking at virus incidence, this is incidence of girdling. And you can see really this pronounced edge effect. It shows up particularly in the block in Southern Oregon, where you can really just see the lighter areas where you're seeing the girdling, and especially there at the bottom, that one side of the vineyard was where you were seeing the most feeding activity. So, and we saw this in other vineyards in Southern Oregon, this really pronounced edge effect. So that leads us to look at the surrounding crop. We were out there beating the bush, looking around, and we did find the tree hoppers. And again, they're on native species like oak, but we also found them on apple and seedling pears. When we look through the literature, we see a number of native species that they're found on: Ceanothus, Madrone, Manzanita, but you also see them here in these orchard crops, walnut, almond. So the news is they have a very, very wide host range. So at the end of the season, we started looking at this oviposition activity. And if you cage these guys on vines, you will get eggs. And the torticellis actually lay more than one egg. They'll lay a series of eggs. And we did find those on grapes, and we're going to follow that, see what happens this spring. And we also put some on other plants, such as pear here. This is a close-up of that earlier photo. And you can see the ovipositor blade sliding in under the bark, and you can see the little oviposition site over to your right, where they put in multiple eggs underneath the bark. And I'll just finish up here. This is sort of our current best management practices. It's evolving, work in progress, and it's really mainly common sense. Plant clean material, make sure you've got clean material. I do want to highlight, you should monitor your vines for symptoms, and if you're out looking for symptoms of virus, you should see symptoms of the activity of the tree hoppers. And, check the edge of the vineyard, look, especially it's going to show up in red cultivars. And this point at the end, really have to be careful planting or replanting vines if you've got red blotch in the area and you have the tree hopper activity. I think the sort of result after finding out what we needed to look for this year, the more we looked, the more we found, really. So sort of obviously ongoing. And I do want to thank the industry, the Oregon Wine Board, special thanks to the collaborating growers, and do want to single out Andy Swan, who was a big help. He's now at the Southern Oregon Wine Institute. And I'll introduce Daniel Sweeney, who's another great collaborator and incredible resource in this research.
Okay, does anybody want to stand up and take like a 30-second stretch break? You all have been sitting for about an hour. Everybody okay? Yeah, just take a moment. So thank you. And does this point? This points? Okay, so I'm going to have to stick to a script here so that I make sure I cover all the information I'm here to present in a timely fashion. Thank you. Here to present on a grower perspective and what I've seen over the last 4 years in vineyards in Southern Oregon. You can see here what Fuchs like to say, welcome to the land of confused people. These are from the fall of this year, this past year, rather. What I'm going to do here is walk you through 2 different vineyards where we've seen sort of what Vaughn alluded to, where you have an intense amount of spread in one vineyard and a slower but significant amount of spread in another. So we're going to go vineyard back and forth between the 2 vineyards. And this is vineyard number 1. This is a production Pinot vineyard with 3 clones, all self-fruited, planted in 2008. And the grapes from this vineyard are all destined for Oregon designate Pinots, so it all comes up here. This is vineyard number 2, again, production Pinot. There are 2 blocks to this vineyard, the lower and the upper. This is the lower block. Nope, went too far. This is the upper block. You can see the details there. This is a relatively young vineyard planted between 2009 and 2012. And obviously, we're dealing with red blotch here. This came in on infected nursery material, as it does. So 2013, this was my first year in the vineyard and first year in Oregon. This is also when we began to take note of red leaf issues in these vineyards. Came from New York, where we had dealt with a lot of leaf roll, but hadn't heard much about red blotch. And you can see here what I was hearing from the research community. Vineyard number 1, we had some red leaf symptoms in the fall. Sent some samples off to Dr. Fuchs, and these samples were taken from across several ranches, but the results we got back were really more leaf roll than red blotch. Okay, can you all see the numbers there? So yeah, 70 plants removed, 20 plants removed. So a small amount, but we removed all the symptomatic plants from this vineyard despite negative test results. When we removed these plants, we were still thinking it was leaf roll, and you can see that more than half of the plants came out of that 115 block. Vineyard number 2, we had a few red leaf plants appear in the fall, and you can see that we did remove block, the Pinot 828 block, and I'll go into detail on that in a minute. Here in the lower block, you can see sort of the symptomatic area. This is an estimate. I was kind of going back in time to do this, but they're along the eastern edge. And we really weren't too worried because research wasn't telling us that it was moving. And the symptoms in that area were fairly sparse in their density. In the upper block, that's the Pinot 828 there in red. And the fruit quality wasn't so good, and the symptoms were more widely dispersed, and so we decided to remove that block and replant the following spring, which we would extend into that yellow section there. So going into 2014 now, this is the state of research again, no big changes. And then back to vineyard number 1, we replanted and rogued plants from 2013 and started flagging that year, painting on the drip line where we found symptoms. And Also sending off more testing, and you can see the results there at the bottom. So here's what we removed in 2014. We still weren't sure exactly what we were dealing with, so we sent more tests off to Cornell in the fall. And it wasn't until these results came back that we realized that we were dealing with red blotch. And also, importantly, that the virus could outpace the symptoms. So here are the test results, 15 total tests. Each group consists of 2 symptomatic plants and 1 adjacent asymptomatic plant, and you can see that everything came back positive. Vineyard number 2, similar to vineyard number 1, we replanted everything that we had rogued, and we expanded the vineyard a bit, and we began flagging in the fall and sent some more testing out. And here is the lower block. You can see this is Vaughn's grid there where the 11 positives are. You can see that the symptoms also spread a little bit, but not a huge amount. This is that vineyard that Vaughn mentioned where we had a field day and we walked out to see. It was those 3 there kind of grouped together. We walked out to see those, and in the process, we'll get to that in a minute. But so the big difference between the 2 vineyards was that we did not rogue at this site in 2014. Here's a map we created with each cell representing a vine. You can see that the concentration of positives, where they're located, is not too alarming. And one detail that we kind of an oversight was we did not, really this was OSU's grid, but did not note the symptomatic status, whether these were red leaf or not. We just took a grid and these are the results we got. So in the upper block, you can see the area we replanted and extended there, the clean material. So at this point in 2014, this vineyard is now basically completely planted, and we're at a net, after removing that 828 block, we're at sort of a net loss in symptoms. And so then we're going to move into 2015. And once again, no big changes in research, but there are some questions about the leafhopper study, which Frank covered. And by the end of this year, some researchers were acknowledging the presence of spread. So in vineyard number 1, again, replanting, flagging, testing, and roguing. And based on the number of symptoms, we made a decision to pull the entire block there. At this point, we knew that it was red blotch. You can see that there were very few new symptoms in the 667 or 777, but we were getting used to the idea of a certain amount of latency, so we were still remaining vigilant there. And we also developed a testing grid with Cornell at this point, which you can see there. There's about 100 vines, more or less evenly across that 667 block, and focus in the area of greatest concern, right? You've got, closer to me here would be where that 115 was removed. So you can see we got 0 positive results, which left us feeling pretty good about our strategy there. But vineyard number 2 was a different story. No replanting there because we hadn't rogued in 2014. Continued flagging and testing, and ultimately ended up roguing a very substantial amount of the vineyard. This is what happened in the lower block. What happened was that by not roguing in 2014, we had essentially left a sizable reservoir of virus in the field. When we began flagging, we noticed that symptoms here had increased about tenfold. Now, I'm talking symptoms. We're not doing vine by vine testing like these guys are, but this is, it's pretty obvious when you're out there in the field what you're looking at. So then we removed every single one of these plants, and if there was a panel, we're at 4 vines to a panel. If 3 of them were symptomatic, we just took the whole thing, or if a row was at 75%, we took the whole row. Here's a plant-by-plant map showing all the gray was what we rogued, so that's a little more precision, the view there, but that was every symptomatic plant in the vineyard, and you can see it was 43% of the block. So here's that plant-by-plant map showing, let's see. These are, remember going back, these are the positives that we had from the year before. So we went into the season Knowing this, just those green, lots of negatives rather, and just a few little positives. And then here were the results from that year. Key detail here is to look at the northern half of that grid, and you can see how well the results coincide with the roguing. Also note the number of negative results at the south end of the grid. So in a pretty heavily concentrated rogued area, there were a lot of results still coming back as negative. So let's see, moving into the upper block. No small amount of infection, but not quite as bad as in the lower block. And remember that everything to the east there is just 1 year old. And here's what we ended up roguing. Take a look at how many 1 year old plants were removed there. So this was our testing there from 2015. Most alarming were the positives in the replanted area, which is that sort of 8 rows, that long, I can't get the pointer to work, but that long, narrow strip of vineyard there. We had 33% positive return in that little acre. And essentially what that means is that we had left a reservoir of virus there to the left, or to the left, to your left. So, one interesting thing to note here is that all those 1 year old plants, let me back up here, all of that same material, we had planted this material in 2 other vineyard sites. So you might say, oh, well, it's coming from the nursery infected, but we had planted that same material in 2 other vineyard sites, and we did testing grids in those as well to the same extent, similar type of testing grid. This was all testing with Cornell, and we got 0 positives in those others. So kind of alludes to the fact that it was coming from this vineyard. So going into 2016, so we have a vector. At least we have one, but of course there could be others. Big challenge with this vector is that, as these guys were saying, they're sort of incidental in vineyards, which makes it very difficult to monitor for them, as you could see by the counts that Rick had. I mean, we know they're here, we know they're moving virus around, but they're difficult to catch and count. So back in vineyard number 1, we're back to a full vineyard, and the plan there is to continue to monitor through the entire season. Sorry. So this is planted out now. We did notice some girdling there starting in June and July, and we also noticed some symptoms appearing mainly in that 667, and we took out about 50 plants from the vineyard, which is more or less consistent with the previous 3 years. And overall, this is what I consider a best case scenario in red blotch management. We identified the presence of the virus, we rogued aggressively, and to date we have sort of stayed ahead of the virus. In vineyard number 2, we replanted in the upper block, but not in the lower. We felt pretty optimistic about our roguing and testing coming into the season, but unfortunately what ended up happening was that, well, you'll see what the virus did. So in the lower block again, we let that rogued area sit fallow. As a reminder, here's where we began the season. Lots of virus and lots of positives, but again, 60% roughly still good, and we're getting no symptoms, no positives in that area. But here's what happened. This map shows the incidence of symptoms plant by plant. Everything in red was essentially latent infection, right? It was beyond the means of detection, either visually or through testing. It had to have occurred in 2015 or before because there were no plants in this gray area during the 2016 season. In essence, we witnessed this block go from 1 positive test result and some sparse symptoms in 2013 to near complete infection by 2016. And of course, you can imagine what it will look like this year. And we didn't know this prior to replanting in the upper block, but we had made the choice to replant in the upper block. And as a reminder, here's where we started the season there. Lots of negative test results, and a relatively small percentage, about 12% of the vineyard had been rogued. And here's where we were by late season there. The light green area is what was replanted. and many of those are already showing symptoms by that very same fall. The red is all newly visible infection and essentially a new reservoir of virus. And you can imagine what it will look like this year. So my advice to growers, I've got a list here to get through. I've got about 1 minute. My advice to growers, buy clean plants, monitor frequently, create maps. It only took me one afternoon to create each of those plant-by-plant maps, so you have the time to do it. If you find or suspect red blotch, look for patterns. Patterns mean it's spreading. Any concentration of symptoms or discernible patterns mean it's spreading. If it's infected nursery stock, it'll have a random distribution. I think Frank did a really good job with the old vines there, just kind of, yeah, it can spread. So take lab test results with a grain of salt. I have literally seen hundreds of tests come back negative, only to have the vines show the infection the following season. As I like to say to Rick, the only test results that matter are positives. A positive means that you have the virus. A negative doesn't really mean anything, at least as far as I've seen. And again, I understand this is an extreme example, but remember that the vector is highly mobile and that the virus can live in wild or feral vines, and be cautious with replanting. Consider an invisible reservoir and learn from the mistakes of others, because it is much more cost effective. Questions that I still have, these are my questions. Why did the virus begin spreading, when and how it did? What do we do with white grapes? I've heard a nice idea of planting some Pinot out there as bellwethers, or red cultivar as a bellwether, which is a really good idea, plant it along the edge. What are best practices for replanting? I mean, my advice is just don't. If you see it, if it's moving, don't plant next to it. It doesn't matter if it's clean, it can get dirty real quick. So we don't really know too well which conditions favor the vector, and we don't really know how to manage for the vector, for an incidental pest in general. Are there other phenological cues or symptomatic precursors that we could look for? And not to open a can of worms, but of course, how does this affect wine quality? We still don't really understand. And Sudi had some work on it, but I think there's a lot of work remaining to be done in terms of understanding whether this is a cumulative impact on fruit quality, or whether you can manage your way out of it. We just don't really know. So with that, thank yous. Mostly worked with these guys here, and I guess that's my time. So thanks.
Well, thank you to all the speakers for being timely and excellent presentations. Good update on red blotch, where we're at, what we know, and a lot that we don't know. I know it's lunchtime, but I'm guessing we can, if people want to stay and ask questions, we can answer questions for a while. Otherwise, you can go stand in queue downstairs and wait to get in line. So if you want to spend 10 minutes asking questions, let's go for it. And the panel will come up, and you can direct them to specific people if you wish. Any questions? Has it been found globally?
Europe, Australia.
Has it been found globally outside of North America? It's primarily a North American virus. It's reported out of Korea and India. Yeah. And there's a report. There's infected plants in Switzerland that went over there from North America. But it appears to be a North American virus that made its way into grapes, and it's primarily a problem in North America that we know of at this time.
Are there some varietals that are more susceptible to this, or do we not know enough about that yet?
I can answer that.
Good.
I think we don't know that.
Okay, that was going to be my answer.
Question over here. What do you do with the infected plants? How do you dispose of them, or what do you do?
We cut them down. Sorry, can you hear me?
You need to turn on the switch.
I think we do. We cut them down, paint glyphosate on the trunk, pull them out in the spring once you can get in, and hope that they're dead and monitor for any regrowth. Burn a pile.
Yeah, and I think you want to be, I mean, this is, how do you manage that is a good question. If you cut those down right at the end of harvest, and you've got vectors on that plant, as they dry up, they're probably being encouraged to move. So, what's the best time to remove? I don't think we know that. And, because if you cut down when there's still foliage on that plant, and you've got insects there, and now that plant is slowly desiccating, the insect realizes that it's not getting any water and nutrition, and it's probably might be encouraging it to move. But we don't know that. So what's the best time to cut those plants? Should we wait till they're fully dormant?
We were doing it on a block-by-block basis. In young plants, we cut as soon as we would see symptoms. In mature plants, we were waiting to get a crop off and then cut them down in the fall. And we would paint glyphosate on the trunk as they were senescing and kind of carbo storing for the winter, and so we're hoping to kind of get some good translocation there.
Right, and you want to make sure there's no suckers coming up before you look at replanting, because any growth from that plant, if you've got a sucker coming up from the root system, it's going to have virus.
Right.
Yeah. So.
Is any of these testing done taking in consideration weak vines and balanced, healthy vines?
I'm not sure.
Any of these testing?
These aren't healthy vines. I mean, yeah, we tested plenty of healthy vines and plenty of infected vines. So is your question whether healthier vines can fight the virus off? Or?
Yes, that is the question.
I don't think so, honestly.
Yeah. I think if you have an infection in June, we don't know if you're going to see symptoms in September or not. From the point of infection, we don't know how long it takes to get symptoms. So you may get a vine that looks healthy that tests positive. It's just that you're not getting symptoms yet.
In my experience, if the vine gets infected this year, it needs one dormant season to show the symptoms, at least one cycle.
I've seen them show up the same year, in those one-year-old plants. But yeah, they did overwinter once. But even in that last slide of the upper block there, we had, and that was Andy Swan who went out with Laura and went vine by vine checking the symptoms, and there were plenty of brand new plants that were.
Okay, I think when you have a vectored transmission, if it happens early in the spring, it is just possible to see the symptoms. Sure. In same fall. That's what we noticed when I showed single shoots where we had symptoms. They had tested negative, those vines.
Right.
Mm-hmm.
Actually, I had two questions. One is over here, Bob. One is, has there been much work on looking at rootstock impact of symptom expression, or maybe rootstocks that might hide expression, which happens with leaf roll sometimes?
There is a trial going on in Davis. Dr. Adib Rohani started in 2013, looking at single sign infected on different rootstocks and single rootstock on different signs too. The results are still, it's grapevines. We need several years before we can get data.
Sure.
Maybe in a couple of years we'll have the data.
One other quick question is have they found the virus in anything besides Vitis? Is there any other hosts that are known?
No.
Good.
We tested several other, like weeds and other things that's in vineyards. They have tested negative so far. It doesn't mean there are we don't, we have excluded everything at this point, but we need more to look at it. Yeah.
It sounded like there were some mixed results in terms of how wine quality was affected. Are there any plans to follow some known infected sites to see what that translates to in wine quality, specifically Pinot in the Willamette Valley?
My understanding is that if you look at in Napa Valley, we get varying results from Cabernet or from Chardonnay. In Pinot, we've heard different results. I think there's a lot of unknowns there. And the other thing is, if you had a growing season like we did the last 3 years, and you can ripen that fruit, it might be just fine, or maybe the impact is minimal if you go back to 2011. So there's a lot of unknowns on that one. And I think ideally, if we want to stress the vines, it'd be nice to be able to control the stress rather than depend on the virus to do it for us. But there's work going on in California and here in that regard. But there's a lot of unknowns.
If I can comment on that really briefly, because this is way out of my area, but Sue and I had an opportunity to go to a winemakers meeting in St. Helena, and they had a panel of winemakers from Paso Robles and Napa Valley that were talking about making wines and everything, and how they deal with the red blotch infected vines. Mm-hmm. And they showed, big differences in quality, but then they were also doing different blends and things like that, and seemed to be able to get by, doing that. So I think there's kind of the whole art in the thing may need to be worked out, but to my knowledge, there's no real good data on that yet.
And it might be that if you have 20% red blotch uniformly spread through the vineyard, maybe that's okay. What's the threshold? And again, there's a lot of unknowns. Yeah.
Question. Both doctors, Hilton and Zalom, mentioned the host range for torticollis. From a practical management point of view, are there other potential hosts in the vineyard, in addition to the grapevines, that we could control to minimize the hospitality, shall we say, of the vineyard? Thistle or other sorts of things that might enable us to decrease the population of these bugs.
I'm in your territory, Rick, if you want to. Okay. No, that's a really good question, and I don't really have any answers for it, unfortunately, but because we're really just working in our first season with this. But we have, with the biology, we know that, we think that it takes a host beside the grapevine for them to develop on, the nymphs and so we know there's a lot of different hosts for that. What we could do to manage that is what we're kind of looking at now. And we actually have plots like this winter where we have cover crop, different plantings of different individual, not mixes, but individual plants planted in vineyards, 3 vineyards in California there. And we're trying to look at, if the vector will overwinter on them and, the success of them, and then maybe what we can do to try to manage those hosts. And so we're really just scratching the surface. But a lot of the work that was done in California on cover crops, I was part of the team that worked on that 20-something years ago. And I think they can be part of the solution, because the first thing everybody said is, do I use Roundup on everything? And no, not until we have a lot more information, because they could be part of the solution then, but I don't know exactly how to manage that or which. So I'm sorry.
So one observation that we made in all of the vineyards in Oregon to date is we've only found adults on vines, no other life stage on the vines themselves. We only found in one situation an exoskeleton of an immature stage on an apple, I think, maybe a foot off the ground, and that's it. So we think that the life cycle needs non-woody plants, and we're going to be focusing on some of those other plants that was listed by Rick.
Yeah, I mean, I think seeing the edge effect, it really tells us it's coming, these insects are coming from outside. And I mean, when you look at the host range, I mean, what vineyard doesn't have oaks near the vineyard? And certainly where we are, we have lots of feral fruit trees and old orchards around.
Oh, right. What Rick
okay.
said this morning was, remember, they're tree hoppers.
Okay, we're going to have to cut the session off. I think the speakers maybe can hang around a bit if you want to come up and ask questions, but it's being recorded, and that part of it's got to end.